All right, welcome to the first lecture of Education and Research Design. Today we're going to talk about the introduction to research design because that will be helpful as you do your literature search for your continuing education project. So research in the clinical laboratory is very important. People with baccalaureate degrees should be intelligent consumers of research. That means the ability to read, understand, evaluate, and apply published research. And since the clinical laboratory is constantly changing and advancing, then we need to be able to read journal articles and understand them and then apply them in practice. So as a clinical laboratory scientist, you should be able to collect and analyze meaningful data and then critically apply research findings either from your own research or from research that you read through journal articles. The rationale for research in the clinical laboratory, that's to develop a knowledge base that maximizes the effectiveness of clinical laboratory science practice so that you can do the best practice possible. So this helps promote the growth of the profession. As we've seen recently with COVID-19, the clinical laboratory has kind of come to the forefront with developing tests for COVID-19. And we've also had to do a lot of increasing the body of knowledge relevant to the laboratory for things like COVID-19 as well as many other diseases. And how do we do that? By documenting and testing elements of practice through rigorous and objective analysis and scientific inquiry. So the purpose of research is, one, to enhance the body of knowledge in the profession. So how much do we know about clinical laboratory tests? To improve our problem-solving skills. Now that you've been in the lab for a while, you should see that things don't always go according to plan. Enhance our standing as professionals as well as experts in the healthcare field. So basically we get more street cred. And then improve the standard of care provided to patients. This is especially important for clinical laboratory scientists as a large chunk of medical decisions are based on laboratory tests. And so those decisions are only as good as those laboratory results. And also by researching, we promote faster change and progress in the profession. So what is evidence-based practice? Evidence-based practice is the principle that occupational practices, choices, and decisions should be based on the best scientific evidence currently available. So as we learn more, and we should apply it. So once you know better, do better. And this can be slightly difficult in actual clinical practice because you have to get your institution on board, but you should be constantly reading the literature and finding out, okay, what is the best practice right now based on the evidence that we have, and then applying that in your job. So study design and data collection, the first thing you have to do is identify the research problem that you want to study. So what is the problem? And then once you've developed the problem, determine the problem, you can help develop a hypothesis or what you think is going to happen. And then you design an experiment to test that hypothesis on a target population. It's impossible to test every person ever, so you choose a target population or a sample subset in order to test. Then you perform the experiment and collect the data. And then you take that raw data and analyze it and then draw conclusions based on that analysis of the data. So a research hypothesis, it's defined as a supposition or proposed explanation made on the basis of limited evidence as a starting point for further investigation. So in general, it's kind of got the formula, if we change this independent variable, so what are we manipulating, then this dependent variable will change, or the outcome. So an independent variable is a variable, this is often denoted by x, whose variation does not depend on that of another. So that's what you're manipulating in the study. So that could be a drug treatment, that could be the type of clinical laboratory test you're using to diagnose a disease. That's what you're changing. The dependent variable, on the other hand, is the variable whose value depends on that of another, which is the independent variable. So this is the variable that that's being observed in the study. So this would be like, okay, if we get cancer patients this treatment, then they'll live longer. So your dependent variable will be how long do they live. Your independent variable will be what treatment are they receiving. The null hypothesis is kind of the opposite of the research hypothesis. So it states that there's no statistically significant difference between the populations or conditions, and any difference that you may see is due to sampling or experimental error. So basically it's saying nothing changed, your independent variable didn't do anything, and it was all just chance. And in order to accept or reject the null hypothesis, you have to use statistical methods on your data to determine whether there was a statistically significant difference or not. And we'll go more into statistics. There's a handout associated with this lecture because you all should have taken statistics, theoretically. So instead of me lecturing about that, I have a handout for you to review if you need it. So the scientific method, it's a systematic, empirical, controlled, and critical examination of hypothetical propositions about the associations among natural phenomena. Basically, you look at the world, come up with an idea of why it's happening, and figure out if that's true. So this definition implies there's a sense of order and discipline that will ensure an acceptable level of reliability. There's organized collection and objective analysis of data. The data must be documented through direct observation to prevent personal bias. And then you must attempt to control factors that are not related to the variables in question. And you need to subject your findings to peer review and scrutiny by other scientists. This is often referred to as rigor and reproducibility, where your data has undergone rigorous analysis, and then it's also can be reproduced by your peers. So some of the assumptions and limitations of the scientific method. The scientific method is based on two assumptions. The first is that nature is orderly and regular, and that the events are consistent and predictable, and that events are not random or accidental, and therefore one or more causes can be discovered. If you've ever actually done research, you realize this is a lie, and that's why it's an assumption. But in theory, things should work the same way every time, in theory. The limitations is that scientific research is not perfect. It's not. I've been through grad school. It's really not. But it is the best we have, so that's what we do. And there's different types of research designs, and they all have their own pros and cons, and they have their hierarchy of what's the best and what's kind of not the best. There's also some extraneous or complex variables that you can't fully control no matter how well your experiment's designed. And this is especially true in human research because humans do what they want. You can't control them. They don't always listen to directions. They don't always give true answers. So you can't control for those things. And people don't live in a bubble. So there are two main types of data. Quantitative data, and this is data that can be counted, measured, or expressed in numbers, so like numerical data. So it's very objective, it's to the point, and it's conclusive. So if we look at our little dresser down here, we can say quantitatively that this dresser is 3 feet tall, it weighs 100 pounds, it has 15 books on it, 3 shelves and 2 cabinets, and it retails for $1,500. So qualitative data, on the other hand, that's descriptive and conceptual. So the data is categorized based on traits and characteristics. It's much less objective than quantitative data. So the same dresser, nothing's changed about the dresser. The qualitative data is that it's made of wood, it's built in Italy, it's deep brown, has golden knobs, smells like oak, and has a smooth finish. Someone else who's looking at it may say something different, and that's why it's more subjective than quantitative data. Another aspect of research is experimental controls, and these help account for extraneous variables, and you want to have as many controls. You want to have controls to make sure that any changes that you're actually observing in your experiment are due to what you've manipulated in the experiment, so due to your independent variable. So having experimental controls strengthens the relationship between independent variables and then changes in the dependent variable. And that helps infer causality. Just because you see a correlation in something doesn't mean that those things, that A causes B. So you have to have correlation does not equal causation. So having experimental controls helps you infer causality. It makes it easier to make that assumption. So research design should have an experimental group and a controlled group. For example, drug studies will have one group that receives the drug, that's the experimental group, and then another group that receives the placebo, and that's the control group. And these groups should be matched for things like age, race, gender. They should be as similar as possible, and the only difference should be one's receiving the drug and one isn't, as much as possible. So there are several types of research. I'm going to go through each of these individually. So there's basic and applied research, experimental and non-experimental research. And you might be going, Dr. Moss, isn't all research experimental? Yes, but no. We'll talk about that more. And then you have descriptive, exploratory, and experimental again. And there are more categories and subcategories that we're not going to go into for our purposes. So this is kind of a picture that I took from an article that goes over experimental design to kind of show in medical research the various levels of research. So you have primary research, and primary research is any time where you are physically collecting data yourself. Whereas secondary research is where you're doing like a review of other people's research or a meta-analysis of other people's research, but you're not actually doing the experiment yourself. And as part of primary research, you have basic research, clinical research, and epidemiological research. And there's subcategories in there as well. The way I'll be discussing them in the lecture doesn't quite fit neatly with this diagram, but the diagram was nice to show the complexity of experimental design. So we're going to talk about the difference between basic and applied research. So research is based on the objective. So which one you choose is based on the objective of the research or the degree of the utility of the findings. So, and by degree of utility of the findings, it means like how useful are these findings. So basic research is done to obtain new information strictly for the curiosity of the investigator. There's no immediate use of the knowledge. So you're just kind of like, I want to know what this receptor does in the cell. Well, why do you want to know it? There's no reason why I want to know it. I just want to know. That's basic research. And there's a lot of people who love to do that. Most medical research is not really going to be basic research. Most medical research is going to be applied research. And that's because it's directed toward solving an immediate problem with a specific application in mind. And with that application usually being directly related to practice. So this would be, okay, I want to know what this receptor does because I want to find a drug that attacks it to help treat this cancer. So you're actually trying to apply the knowledge to some sort of clinical practice. Now, experimental research, this is research where the researcher manipulates and controls one or more variables and then observes the results. And this can suggest cause and effect relationships. So the researcher is actually going in and changing things and keeping other things controlled. So they're manipulating and they're controlling. They have controls. They have experimental group. This is an experiment. Whereas a non-experimental or observational research is where you're just you're observing. You're just looking. So this generally refers to more descriptive or exploratory investigations where you're just like, let me see what these monkeys do in the wild. You're not changing anything about the monkeys, you're just watching them. And this doesn't exhibit any direct control over the variables, you're just watching. Descriptive research, this is an attempt to describe a group of individuals on a set of variables. So this can use both quantitative and qualitative data. So you could be like, okay, my group of individuals are black, and they're all about 6 feet tall. This is very useful for identifying characteristics, frequencies, trends, correlations, and categories. And you can collect this data in a variety of ways, such as questionnaires, interviews, direct observation, but you can't control all the variables. And this may create a basis for future research by identifying a problem. Exploratory research, this examines phenomena of interest and then explores its dimensions and how it relates to other factors. So it helps search for relationships and predict correlations, and it helps develop better understanding of an existing problem, but doesn't actually come to any conclusive results. You're just kind of getting a feel for the field and figuring out kind of what's going on, but you're not making any conclusions. You're just exploring, seeing what's out there. This type of data research will have mostly qualitative data, such as things from surveys, observation interviews, focus groups, or literature searches. And then we have experimental research. So this compares 2 or more conditions, and it uses controls to account for extraneous factors, and then it provides the greatest degree of confidence in the validity of the outcome, and it usually obtains quantitative data. So this type of research, because you're controlling and manipulating the variables, you have the greatest idea that the independent variable affects the dependent variable, and it's not some sort of extraneous variable that wasn't accounted for because you're setting up the situation. So the research process is a systematic process of sequential steps for thinking, planning, and analysis. So you just don't come up with an idea and just do whatever you want. You have to have a process on how you're doing it. So the first step is to identify the research question, and that may seem easy, but it's actually really hard. Then you have to design the study in such a way that it will answer the research question. You have to figure out your methods, and whether your methods are actually feasible or not. Collect the data, analyze the data, and then draw conclusions and communicate those findings to the broader world. So the first step in the research project process is identifying the research question. So you need to formulate a specific research question. If it's too broad, you would have way too many experiments. It would just be extremely difficult to plan an experiment that would answer a broad question. So it's very nice to have a specific research question so you can really hone down on what your method should be and what your hypothesis should be. So you want to identify the type of individual to whom the result will be generalized. So what sample are you going to need in order to generalize it to a particular population? You need to conduct a literature search or review to kind of figure out what the current theories are so you can make a hypothesis. And then the rationale for the study, you need to come up with that. And the rationale is the justification for why you're conducting the study. Why is it important? Why is it important that you spend all this time and all this money and all this effort on this study? Because you have to get grant funding and it needs to have a reason why you're doing it. And then you propose your research hypotheses based on all of the effort you've done previously. So the next step is designing the study. So in designing the study, you come up with what methods you're going to be using. So how are you going to select your subjects? How are you going to test them? How are you going to measure the outcomes of those tests? So in this part, you're choosing your research method and how you're setting up the entire design. It can actually be quite complex because there's so many different ways to do things, and it really depends on what question you're asking and what you're going is going to lead how you're going to do it. So it's important to clearly define all the methods and interventions. What are your independent variables that you'll be manipulating? How are you going to manipulate them? What are your controls going to be? How are you going to ensure that they're actually controls? How you can make sure there's not extraneous variables that you haven't accounted for? What dependent variables are you going to be monitoring? How are you going to monitor those? So there's a lot of logistical questions involved in designing the study. So the next step in the research process is actually doing the method and collecting the data. So before you do this, you have to get your plans approved, especially if you're doing human research. You have to go to an institutional review board to make sure that your research design is ethical and get it implemented. This is usually the most time-consuming part. Some of these studies can last years before you reach the end point of your study. And then you take your raw data and collate it for analysis. So the next step in the research process is actually analyzing all that raw data that you collected over however long it took. And you need to analyze the data using statistical procedures and then interpret the data and draw conclusions from that data and determine whether your research hypothesis was supported or rejected, and the same for your null hypothesis. And then last but not least is to communicate your findings. So researchers have a responsibility to share the information that they find, whether that's via a journal article, an abstract, oral presentation, or a poster, so that it can be peer reviewed and undergo the rigor and reproducibility test. So in your communication of your findings, you should include any known limitations of the study. That could be as simple as we weren't able to study a lot of females. We weren't able to get a lot of patients for this. So whatever the limitations are. And then offer suggestions for further studies. And usually as you're doing research, you kind of go, oh, I saw this result. I should study this other thing because that ties in. I wonder if that's what happens. So usually it's very easy to come up with suggestions for further studies. So our retrospective research designs, this is kind of a broad category of research designs, and there are several studies that we'll talk about that fall under retrospective. And a retrospective research design is any study that looks back in time. So a lot of times we'll be looking at exposures to suspected risk or protection factors in relation to an outcome that has already been established. So this would be people that say, these are people who develop lung cancer. So you look at a group of people that have lung cancer and a group of people without lung cancer, and you go, what exposures did they have? Then you'd have a theory on what exposures you're looking at, so such as smoking. So you're like, okay, out of these people who have lung cancer, how many smoked? And you'd have inclusion and exclusion criteria that are necessary to minimize bias. So the retrospective study, you have your outcome. So these are patients that already have the outcome desired, and then you're looking back in time as to whether they received an exposure that you think is the cause of that outcome. So because you're looking back in time and it's impossible to get a person's full history, there are lots of opportunity for confounding bias, confounding and bias errors. And it's also, what's nice about these studies is that it's very quick to get results because these people already have the outcome. You just need to figure out if they've had the exposure. And it's relatively inexpensive because the data is already available. Prospective research designs, on the other hand, this takes a group of people who have received the exposure that you're interested in, and then you're going to follow them over time to see if they develop the outcome. So you're looking at usually two different patient populations, one that's received the exposure and one that hasn't, and then following those two groups and seeing how often each group gets the outcome. So this would be for our smoking study. You look at people who are currently smoking and currently not smoking, and you'd see over the next 10 years how many people in each group develop lung cancer. So this usually takes a cohort of subjects, so your groups, and watches them for a long period of time. This is very time consuming because you might be looking at the same group for 10 years, 20 years, 5 years. It could be very long. And it's expensive because of that. You also lose a lot of people to follow up. They don't respond to your messages anymore. And it does have fewer potential sources of bias and confounding variables in retrospective studies, but it's more expensive and time consuming. So if you want to publish something quickly, sometimes a retrospective study is just easier, but not as rigorous. So now we're going to talk about the broad fields of medical research: therapy, diagnosis, screening, prognosis, and causation. And each of these fields of medical research have research study designs that are better suited for them, and we'll talk about each one. So our first broad field of research is therapy. And therapy research, you're going to be testing the efficacy of drug treatments, surgical procedures, alternative methods of service delivery, or any other intervention, and seeing what the outcomes are. So the preferred study design, the gold standard, is a randomized controlled trial. A randomized controlled trial is where the investigator randomly assigns the people in the study to an intervention group, so this would be the treatment group, or to the control group. And in the treatment group, you receive whatever the intervention is, whether that's a drug or a procedure or additional intervention. And then the control group, they can either receive no treatment whatsoever, they can receive a placebo. A placebo is preferred over no treatment because then you can kind of see if there's a placebo effect, or the standard of care. And that control group is that's where your institutional review board comes into play with determining the ethical considerations. Because if you have a patient population that has leukemia, it's not really ethical to not treat the patients who have leukemia in the control group. You have to give them something, otherwise they're going to die. So the control group might receive the normal chemotherapy that people with that type of cancer receive, whereas the treatment group will receive the new drug. And a randomized controlled trial presents the strongest evidence for causality, and these can be set up as blind or double-blind strategies. A blind randomized controlled trial is where the treatment and control group people, the actual patients in the study, don't know what group they're in. They don't know if they're getting the placebo or if they're getting the treatment. A double-blind study is where the researchers also don't know, and that helps prevent bias from the researchers. And there's a third party who's in charge of determining, of analyzing the data and determining. Our next broad field of research is diagnosis. This is where the clinical lab has its heyday. That's where they're big. And that's demonstrating whether a new diagnostic test is valid or reliable. So this could be things like laboratory tests, or it could be things like so the preferred study design for diagnosis studies is a cross-sectional study in which both the new test and the gold standard test are performed side by side and compared. So a cross-sectional study, this is a type of observational study that analyzes data from a population at a specific point of time. So this is or a snapshot. So there's no follow-up period, which makes it less expensive than a longitudinal study or a prospective study. It's usually very fast because you're doing that snapshot, you do the test, you do the comparison, you're done, you publish. But it's unable to establish causality. And the major purpose is to describe the frequency of a disorder within an identified group or to compare two groups at that brief moment of time. So you have your study population and you divide them up into no disease and disease, and then determine, for our purposes, be like, okay, was the gold standard positive? Was the test method positive or negative, in comparing them? Our next broad field of research is screening. So this demonstrates the value of tests which can be applied to large populations and which pick up disease at a pre-symptomatic stage. So as you've probably learned in clinical chemistry, is that the prostate-specific antigen tests is FDA approved for screening for prostate cancer, but there's not really any other markers that are approved for screening for cancer and other types of cancer. So we also learned, you might have also learned that colonoscopy is a good, or a fecal occult blood test is good screening methods for colon cancer. So when you're trying to develop a screening test, you need to do a cross-sectional study, which we just talked about, where you take your two populations and your two tests and then look at them at that brief snapshot. And you can also do it longitudinally, where you look at, okay, let's test them at this time period and follow them for 5 years. And the people who had a higher level of whatever your test analyte is, did they develop cancer in the next 5 years or not? So this might be a little bit more time consuming, and it might be longitudinal. So your next broad field of research is prognosis, and this is determining what is likely to happen to someone whose disease is diagnosed at a particular stage. So for example, what's the 5-year survival in a particular type of cancer? And you can develop that based on what treatment arm are they in, what treatment did they receive, and this would be a longitudinal cohort study because you do need to follow them with prognosis. So cohort studies, a cohort is a group of individuals who are followed together over time. So for example, your class is a cohort. You are all going together, we will be following you for our NAACLS data together. So you are a cohort. So in a cohort study, the researcher selects a group of students that do not yet have the outcome of interest and then follows them to see if they develop it. And then selection is based on exposure to a particular agent, such as a vaccine, a drug, an environmental factor, a drug or an environmental factor. So your cohort, you'll have a cohort that's been exposed, and you'll have a cohort that's not exposed, and you'll follow both cohorts and then determine if the disease develops or does not develop in each one. If you just did exposed, then you wouldn't be able to compare them to the not exposed. You need both groups, because otherwise you might find out that the disease actually develops more in people who weren't exposed versus the exposure group. So you need both groups in order to compare them to make sure that you can develop infer that causality of your exposure to the disease. So the next broad field of research is causation, and this is determining whether a putative harmful agent, such as an environmental pollution, is related to the development of an illness. So the preferred studies designed for these is a cohort or a case control study, and which one you use depends on the rarity of the disease. And if it's very rare, you might need a case report. So a case control study is where you have a group of individuals that are selected on the basis of whether they have the disorder of interest. So this is used more often when the condition that you're testing is rare. So the cases in the case control study are the people who have the disorder of interest, and the controls are chosen as your comparison group, and they don't have the disorder, but otherwise they're pretty well matched to your cases in terms of demographics. So this is usually a retrospective study of both groups with respect to the exposure histories, risk factors, and it's usually the only way to study rare conditions, because you can't really do a cohort study for rare conditions because you would just need so many people in your cohorts in order to get enough cases that developed. So if it's something like diabetes, a cohort study is fine. Lots of people get diabetes. If it's something like this extremely rare case of brain cancer, then you're going to need to get the people who already have the cancer and then look back at their exposures, just because you'd have so many patients in your study otherwise. It would just be insane. And there is bias present in choosing what is considered a case or not a case. And then you have a case report or a case study. These are usually published when you kind of have a patient who doesn't present the normal way with the disease. So these are descriptions of interesting, new, or unique cases. So these involve an in-depth description of an individual's condition or response to treatment. It can also focus on a group or an institution or a social unit. And it usually emphasizes unusual patient problems or diagnoses that present interesting clinical challenges. So this would be like someone who has leukemia, but not the normal type. Like this would be someone whose leukemia didn't read the textbook, and it's presented some sort of clinical challenge or something very unique that most people aren't going to be able to see at their institution. So that you write a case study on it so people can read it and be like, oh yeah, that was super cool. If I'll know to look for that now. So these are very rare cases and very unique cases that get case studies written by them about them. Meta-analysis is actually done on secondary, it's this type of secondary research where the researcher isn't actually doing their own experiment. So this combines the data from a ton of studies that all tried to answer the same research question, and usually these meta-analysis is done when these studies came to different conclusions. And the purpose of the meta-analysis is try to come to one definitive conclusion. So this is more than just a summary of the different articles. This is actually taking all their data and combining it using statistics, and you often end up with a plot like this where you have the different studies that answered that question and then seeing what their results were. So you usually get this box and whisker plot, and it compares them, and you can kind of see what the mean of the groups of the studies were, the differences in the studies in terms of their dispersion and things like that, and it really gives you an opportunity to compare them and combine them. And this is useful for increasing the power of the study by increasing the sample size. It improves the estimates of effect size, resolves uncertainty, and improves the generalizability of the findings. So this is considered one of the, next to like a randomized control trial, this is one of the this is what clinical practice is going to be based on. What is the definitive answer on best practice? So there's a hierarchy of evidence where there's different types of primary studies, and they carry different relative weights when they're being used to make decisions about clinical practice. So right now I have it ranked from most reliable to least reliable. So a systematic review and a meta-analysis, though technically these are secondary research, they're considered the most reliable. Then you have randomized controlled trials, cohort studies, case control studies, cross-sectional studies, and last is case reports. And case reports are the least reliable because it's usually just one case. It's like very low power. So now we're going to talk about ethical issues in clinical research. So these ethical issues include the need to conduct meaningful research, the protection of human rights and dignity, the ethical dilemma of control groups, and then informed consent. And we'll talk about each one of these individually. So the first ethical issue is conducting meaningful research. So this is where the rationale for your research comes into play. So this is why you need to justify your research or your project based on the scientific value of its results. So why are you doing it? This is especially true if you're going to be using human subjects. It's not ethical to do a research project on human subjects just for the fun of it. There needs to be a reason why you're doing it, a justifiable reason. So you need to base your research on rational theoretical principles and use sound research design, because you don't want to recruit people into your study and not have a well-designed study and it just be a waste of everyone's time and potentially harm your patients for no reason. And you also need to minimize personal bias when you're doing it. So the next ethical issue is protection of human rights and dignity. So when you're using human subjects, they're humans. You can't do things that are unethical to them. They need to have their own personal autonomy. Anything you do to them needs to be with a sense of beneficence, so you don't want to harm them. Anything you do needs to be for their benefit. And there also needs to be justice involved. This is why there's institutional review boards. And when you get institutional review board training, you learn that you can't do clinical trials on prisoners. You might be like, oh, but they're just this patient population waiting to be used. You can't do that. That's not ethical. You can't really, it's very hard to get IRB approval to do clinical trials on children, on pregnant women, because the risk is too high, or they're at a vulnerable population. So you have to protect people's human rights and dignity and not take advantage of them. And then there's the ethical dilemma of control groups, which I touched on briefly earlier. So with a randomized controls trial, you have your experimental group and your control group, where the test group receives the treatment of interest and the control group does not. But if someone has a deadly disease, it's not ethical to withhold treatment from them. It's not. So you have to give them something. So sometimes if a person has a terminal illness, they will get the standard of care, whatever that may be. The test group might receive the standard of care in addition to the treatment of interest, just so in case the treatment of interest doesn't work, they're at least getting the standard of care. And if the study is going really well and the treatment in the test group is getting fantastic results, the IRB might step in and during the clinical trial and be like, hey, your drug is so good that it's not ethical to hold it back from the control group. At that point, you're like, yes, my drug is awesome. So the results of a clinical trial are constantly being monitored to make sure that the drug that you're testing isn't super harmful and having worse outcomes than the control group in terms of like death and side effects and things like that. And also, if it's doing super well, they might start giving it to the control group because it's not ethical to withhold it. So there's a lot of ethical concerns with the control groups as well as the experimental groups and making sure that you preserve that human right and dignity and do what's best for them. And then we have informed consent. So anytime you're using humans for research, that person needs to have the ability to agree to participate with full understanding of what will happen to them. So they need to understand all the potential risks and benefits. They need to know what procedures they're going to be undergoing. They need to know the side effects potentially, how the information that's collected from them will be used. And you can't just write that in science jargon. It has to be written in lay language and given to the subject in written form. And that information that you're giving to them is approved by the institutional review board before you give it to them. And the consent must be voluntary. They must 100% understand what they're signing up for. And then with the ethical dilemma of informed consent, if you tell them everything, that might actually mess up your study. So by biasing the subject. So the IRB or Institutional Review Board will approve withholding information at times as long as the subjects are being told that information is being withheld. So those would be things like randomized control trials where you're blinding the groups, where you would basically be like, hey, we're not telling you what group you're in. So you are withholding information, but that's in order to make sure that there's no bias in the study because that would mess up your study. And then another part of research is subject information that should always be kept confidential and anonymous. Subjects are told the steps being taken to ensure the confidentiality and anonymity of their information, and they are also told if they're being recorded, photographed, or videotaped. So a lot of times when you see case reports, you'll see pictures like this one to the right, where the person's face is obstructive so you can't identify who they are, but you still get the relevant medical information from the picture. For in this case, we can see that she has a rash on her face. So the Institutional Review Board, or the IRB, that's an administrative body established to protect the rights and welfare of human research subjects that have been recruited to participate in research activities. So each institution that does research has an IRB, and any researcher who wants to do research on human subjects has to take IRB training and then submit a proposal to the IRB and get a ton of paperwork, and everything has to get approved before conducting any research involving human subjects. And this includes analyzing data that's been previously collected. That's pretty easy to get IRB approval for. You just be like, hey, so I already have this data, like it's just sitting in medical records, or I already have it, can I now use it for this new thing? And they usually be like, oh yeah, there's no like harm to the patient. So usually they approve that pretty easily. But something like a clinical trial goes through many iterations and before it gets finally approved by the Institutional Review Board. So research on previously collected specimens and data. This offers a lot of potential for significant discoveries, because if you come up with a research question, you can be like, hey, let me look over the last 10 years of medical records and see if I can answer this without doing a prospective study. Can I do it retrospective? This data is already sitting there in the medical records. I just have to find it. This is called data mining. What's nice about this is that there's no physical risk to the patients, because all this data is already there. It's already been taken. It's already available. Or if you already have the specimens and they're sitting in a freezer, all you have to do is analyze them for whatever your analyte of interest is. There are some ethical concerns with this, is that these subjects haven't given consent for their data or samples to be used for that study. There may be breaches of confidentiality that may be concerned. Certain groups may be harmed based on the result of your study. So you might have genetic research that IDs someone as being more likely to commit a crime, that they're going to develop a disease. Do you have an ethical, are you morally obligated to contact that patient and tell them, hey, you have this gene and you're 100% going to get ovarian cancer in 5 years? Do you have to contact them? Do you even have the ability to contact them? That kind of thing. And then there's also the concern that since the subjects aren't giving their consent, they aren't being allowed to say, no, I don't consent to that. Because they might have a particular reason why they don't want to participate in that type of study. They might not want their sample used for a particular type of research for religious reasons or other moral reasons. So here are the references that I used for this.